All right, so now that we've spent some time talking about DNA structure, we're going to start moving into why DNA is so important. So we've talked a little bit about what it is and why it looks the way it does. But now we're going to start talking about why we care so much about DNA, why it has this reputation, right? And so it's going to take us once again to the central dogma of biology. So this is like it in physics you have, the laws of thermodynamics, and in chemistry you have those laws as well. And here in biology you rarely see laws that are so absolute. It's pretty rare for us to have a thing that happens every single time because living things are super dynamic and so they change like crazy. It's really hard to come up with patterns that span all living things. But this is one thing that does span all living things as far as we've seen. DNA holds information, and then we transfer that information into the form of RNA, and then the information in the form of RNA is translated into a protein. So remember chapters 5 and 8, why proteins are so important. They do so much inside of living things. And I like to think of DNA as being a cookbook. I like to think of it as a cookbook that's in a dead language, like Latin, right? So it's a cookbook that's in Latin, and I don't speak Latin. I, as a cell, don't know how to use the DNA. So instead, what I'm going to do is I'm going to transcribe it. I'm going to get somebody who can speak DNA, can speak Latin, transcribe it into English for me. So RNA is kind of a more usable form of that information. And then I'm going to translate it into a protein. So I'm basically taking the information, I'm going to transcribe it into English, and then I can actually use the recipe. I can actually make whatever it is that the recipe codes for, right? Whatever it is that the cookbook shows us. Okay. So the first process, so we're zooming in here on the first process, which is transcription. So going from DNA to RNA. And some of what you're seeing here will look familiar to what we saw during DNA replication. So like we see that we have a DNA molecule, right? Those are the blue strands here. And the DNA molecule is kind of being opened up, right? Like we saw with DNA replication. And you're going to see that DNA needs to be opened up anytime you want to do anything with these bases. Anytime you want to do anything, you need to open up the DNA in order to access the bases, right? And so we are going to see an enzyme called RNA polymerase. So we've actually seen DNA polymerase before. So if you'll recall from chapter 16, that DNA polymerase is the enzyme, because it ends in ase, that polymerizes DNA. So now this is going to be the same thing, except instead of it being DNA, it's going to be for RNA. So basically, this is the enzyme right here, RNA polymerase, that is able to take that DNA information and transcribe it into RNA. This is our translator. Okay. And so basically what's going to happen is the DNA is going to get opened, and then the RNA polymerase is going to use a piece of that DNA as a template. And they're basically going to create a copy. So it's almost like DNA replication. Remember, like with DNA replication, we had RNA primase come in and it made a primer. This kind of does something similar. It makes a complementary strand, right? Just like we saw before, except this RNA molecule is a little different from the primer we saw before. Firstly, it's longer, but it's also going to have an entirely different job. So, again, RNA polymerase is very similar to DNA polymerase. So we once again have an issue where DNA polymerase and RNA polymerase can only go in one direction. They can only go from the 5' to the 3' direction. Now, I don't need you to worry too much about being able to recognize 5' and 3' directions, but I do care that you know that there are different directions and that there are limitations to what these enzymes can do. So, for instance, this DNA strand right here is made up of a red side and a blue side. And depending on which side we try and go on, so like the red side's 3' is over here on the left and the 5' is on the right, whereas the blue's 5' is on the left and 3' on the right. Depending on which strand we want to use as our template, that's going to affect whether RNA polymerase can go in the right direction. So what you'll see is happening here is the red strand, right, is the one that is the template. strand. So basically, we're opening up the DNA. So now we have two strands to choose from. We have the blue one, we have the red one. RNA polymerase can only go from the 5' to the 3' direction of the strand it is making, right? And so it needs to create an anti-parallel strand. So if the red strand is going from right to left, right, because I have 5' to 3', then that means that the complementary strand is going to be anti-parallel, and so it's going to go from left to right. And so you're going to notice that they still have that anti-parallel quality that we saw with DNA replication as well. So you are going to see a few different terms here, like the sense strand, also known as the coding strand, the anti-sense strand. Your book will talk about a lot of different language, and you don't really need to know any specifics on that, so I don't want to get too bogged down on that. Basically, one of the strand gets used and one of the strands doesn't, right? Because one of the strands, the non-template strand, is harder to use because it's in the wrong direction, right? All right, so basically that RNA polymerase is going to make a copy of RNA. It's going to make this blue RNA strip here from that information. And now we have this RNA strand, and it's going to be its own thing. It's not going to hang out around the DNA. Okay. So RNA is totally happy being single-stranded, whereas DNA has to be double-stranded, like for long periods of time. So like here, DNA, it makes that double helix every time. But RNA is totally happy being by itself. Okay. And just also a reminder, there is no thymine, no T in RNA. There is a T in DNA, but instead of a thymine in RNA, we have a uracil, a U. Okay, so that's just a reminder from chapter 16. And so you need to be able to, given a DNA sequence, figure out the complementary RNA sequence. So you need to be able to tell me, like, okay, here is the DNA sequence. Now, what RNA bases will match with these DNA bases? So imagine that you are RNA polymerase, okay, and we have a 3' 5' here, so the DNA strand is going in this direction. So we're going to be synthesizing our RNA strand from left to right, right? Because we go from the 5' to 3' end. So what matches up with A? So it might be tempting to say T, right? Because that's what you've been trained to do. But in reality, we're making RNA, and there is no T. There is no thymine in RNA. There is instead a uracil. So it's going to be a U. Now, what matches with T? Well, A matches with T. What matches with T? A. What matches with A? Well, a U. Oh, I'm sorry. I just made the mistake of writing a T. Oops, I said the right thing, but I wrote the wrong thing. What matches with A U, etc., etc., etc. So you need to practice recognizing whether you are synthesizing DNA or RNA, and you need to be able to create an entire strip based on a template strand. So I have given you a template strand in the case of this diagram, and then you need to be able to synthesize the complementary RNA strand, okay? As if you were RNA polymerase. So when you actually make that RNA strip, sorry, I've got a bad cough right now. When you actually make that RNA strip, it isn't quite done yet. Okay, so we call it pre-mRNA. So this is what happens with most organisms. Most organisms, so like anything more complex than a bacterium, for our class, is going to do this. They're going to need to change their RNA a little bit, because like right now it's called a pre-mRNA. So basically, like this is the exact code that they synthesized, and that's it. But in order to mature our mRNA, we need to add a cap to one end, and that's going to be the 5' end, and I'll talk about that in a minute. And we're going to add just a bunch of A's, just a bunch of adenines over and over and over and over and over again at the end. So that's going to be at the end of the mRNA, so the 3' end. And so we're going to call that a tail because it's at the back. Okay. So basically, we just need to do these things in order to protect the mRNA and also to allow it to do its job. Okay, and so you'll notice that in this diagram, there's a thing called introns. So introns and exons are opposites. So the mRNA basically has a bunch of information because your DNA strand was really long. And so some of that stuff is important and some of it isn't. And so the introns are going to get excised or removed. So we're going to cut out all of these introns so that we have just the exons. Okay. And that's going to become clear as we kind of go through it. And again, you add a cap, and then this will actually allow the ribosome, remember that ribosomes are responsible for synthesizing proteins, to attach to the mRNA molecule. So basically, I have an mRNA molecule, that's right here in the red, that I have synthesized from the DNA. Okay. So I took the DNA information, I changed that information into a different form. The DNA molecule is still there. I did not ruin the DNA molecule. I just took that information, that sequence, and I have copied it into mRNA. So that process right there was transcription. Okay. Transcription. And then this mRNA, that's the red strip, is going to leave the nucleus and it's going to head towards a ribosome. Okay, so that's what this blue amorphous blob is. And as you guys remember from chapter 6, ribosomes are responsible for making proteins. So this purple bead-like shape that it's making, that's a protein. Okay, and so all of these instances where I'm talking about ribosomes and everything, remember that those are all organelles that are only present in eukaryotic cells. So just remember that prokaryotes, so like bacteria, for instance, and archaea, for instance, they have their own stuff that's a little different. And so I'm showing you the more complicated version, which is eukaryotes, and then prokaryotes would be simpler. They wouldn't have a nucleus for transcription to occur in, right? Because they don't have any organelles. Okay. And so that takes us, now that we've made our mRNA strand, that takes us to the second piece of gene expression of the central dogma, which is translation. So now we're actually going to make the protein that matters. Okay. And so again, I had my DNA molecule, I transcribed it to make mRNA. So that's right here. This is my mRNA. And that's a transcript, right? So I took the information, I copied the information. I didn't do anything to the DNA. I just copied the information into an mRNA transcript. And then that mRNA transcript needs to make its way towards a ribosome. And that's where translation is actually going to occur. So translation is where we actually make the protein. And so basically what's going to happen is the ribosome is going to latch on to the mRNA. It's going to find a specific spot on the mRNA that it can grab onto. So it is not just any old spot. It is a specific place where it can grab. Okay? And there are chemical reasons for that you don't need to worry about. And it's basically going to drag the mRNA strand through it. Okay? So the mRNA strand is going to like feed through it. And slowly over time, the ribosome will be adding amino acids. So each of these little dots are amino acids. And then once it's finally done, we have one large polypeptide or protein. Okay. So there's a specific place where it's going to start. It's going to go all the way through. And then there's a specific place where it's going to end. Okay. And so basically the way that this happens is ribosomes are made up of two parts. You don't need to memorize the two parts or anything. But basically, if you look inside of the large part, so the large ribosomal subunit, this top part here, okay, there are three little spots, three little windows, if you will. Okay. And those have different names. Like we have given those spots names, and you don't need to worry about that. But basically, they're little windows where tRNA molecules can come in. So we had mRNA. M stands for messenger RNA because it holds a message, right? So that's what we made from the DNA. Okay, so we have mRNA that holds a message. tRNA is gonna stand for transfer RNA because what they're going to do is basically bring an amino acid. So that's what this little dot here is. They're going to bring an amino acid to the ribosome so that the ribosome can actually start to string together amino acids. Okay. So the ribosome is going to clamp on to the mRNA transcript. I've just circled the mRNA transcript here. It's going to clamp on, and there's a specific tRNA molecule that's going to pop onto the start. So here, right here, we have an AUG that was on the mRNA molecule. AUG is a start codon. So codons are basically little groups of three nucleotides on an mRNA transcript. So like this mRNA transcript has a lot more information on here than you're seeing, right? Here you just see AUG, but in reality, there's going to be more letters after. Okay, so they're always going to be in groups of three. So this right here, this AUG, that's a start codon. Okay, so that's going to tell the ribosome, hey, start here. And then this next codon is going to code for something else, and we're going to get to that in a second. And then this next codon is going to code for something else. Okay, so the mRNA transcript has codons, which are little groups of three nucleotides, whereas the tRNA has what we call anticodons. So it's like the opposite. Okay, so here we have complementary bases happening again. So basically, if the mRNA has an AUG right here, then the tRNA that's going to come in needs to match those with complementary base pair rules. So the tRNA is going to have a UAC. Okay, so it's just going to latch on there, and then it's going to pass the amino acid. And we're going to model this in class, so I don't need you to freak out about it. But just as a little primer, if you will, here's our RNA molecule. And if we look at all of the nucleotides and we break them into groups of three, we call those codons. And those codons are going to code for different amino acids. So if your codon says GCU, that is going to, when you translate it, change it into alanine. Whereas if your codon says ACG, that translates to threonine. Okay? And so these are all different amino acids, right? And they're going to string together and form a polypeptide. Okay? And so this is a universal code. Every living thing uses this code. And so you're going to see a couple of different styles of tables. All right, so the first style of table you need to be familiar with will show you first nucleotide position here. Okay, and so on the left side. So what I need to do is take the first nucleotide, G. So I'm going to take the mRNA codon, and I'm going to look at G. So I'm looking at this row right now. Okay. My next nucleotide in that codon is C. So up here I can see that my second nucleotide position is determined by the column. So I'm going to circle this column. Okay. So basically, I need to figure out what amino acid will be coded for by a GCU. So I did my G, that told me it was in this bottom row. I did my C, that told me it was in the second column. And a GCU is alanine. Okay. And so that's how I figured out that this was alanine. And so you could go through and do this for every single codon on an mRNA strand, and we're going to practice with that. But there's a couple of things to notice here. A lot of codons will code for the same thing, right? Like there's lots of ways you can make an alanine. Alanine can be a GCU, or it can be from a GCC, or a GCA, or a GCG. Right? There's lots of different ways to code for the same thing. And that's because look at how many different combinations there are, right? There's 4 in every box, and there's 16 boxes. So there's 64 different combinations of those 3 nucleotides on the codon, within a codon. And there's only 20 amino acids. So there's going to be some overlap, right? And so you need to be able to recognize where a codon chart will take you. So like if I give you a codon, you need to be able to translate it into the appropriate amino acid. If I give you a codon chart, you do not need to memorize the codon chart. That is a waste of your time. Do not memorize this chart. It will be provided to you. On the actual AP test, you are given a codon chart. Okay. So basically, if you're going to use one of these tables, you need to find the 3 bases of the codon. All right. Generally, if you're using a table like that, you're going to see that the first base is on the left-hand side, and then the second base is along the top, and that'll tell you like which box you care about. And then finally, you can figure out which codon you care about once it matches inside of that box. And there are other forms of this table. I personally don't love the box form. I really like this one circular one that we're going to use in class a couple of times, but you should just be prepared for any of these. And here's some practice problems. So if you want to go back and take a picture of the codon chart, that will allow you to practice with this. We're going to do this in class as well, but it's really important that you're able to translate a codon. Remember, codon is on the mRNA strand into a string of amino acids. Okay. And so if I were to do that, I have here a codon. Okay. I know it's codon because it's mRNA, and I can translate it into a string of amino acids. And you'll notice that every string of amino acids eventually ends in a stop. And the stop will basically just tell everything to go home. Basically tells the ribosome to let go. And it tells the polypeptide to kind of go off on its own. Okay? It just makes everything dissociate or go away. Okay? So, just a reminder of terminology. The mRNA strand, so this is the thing that we transcribed, has the codon, right? So the 3-letter codon. Whereas the tRNA, so that's what's being shown here, the tRNA molecule has the opposite of that, the anticodon. And so those are going to match up. Okay? So there's tons of these tRNA molecules all over near ribosomes. And so they're just going to be floating around. And if they happen to find the right sequence, so the right group of 3, so the right codon, then they're going to latch on for just a second. And that's just long enough for the ribosome to grab the amino acid and put it onto the chain. So you are going to see this codon a lot. This is the start codon, AUG, and it codes for methionine. Okay. And so that's going to be at the start of every polypeptide, of every protein. One thing that a lot of people do is they accidentally take the anticodon and they put that into this chart in order to try to figure out what amino acid to put. Okay. You always put the codon in here, not the anticodon. And I know it's annoying that you have to remember that. I just always remember to call this thing a codon chart. Okay? And then that tells me like, oh yeah, I used the codon for it, not the anticodon. Okay? So you only care about the anticodon if you're thinking about the tRNA molecule. So that's this whole thing up top. If you're doing any translating or anything, you use the codon. Okay? And so basically there's 3 different spots on that ribosome. Like I said, 3 different windows. There's an E spot, a P spot, and an A spot. You don't need to memorize that. Okay, basically the tRNA molecules will enter in one side. They'll actually add their amino acid in at another spot, and then they'll leave in the E spot. Okay, and so it's basically just this ribosome is effectively moving to the right along this mRNA transcript. Or you could think of it as the mRNA is being fed through to the left. It doesn't matter which way you think about it. Okay, and so eventually at the P site, so the P site is where they're actually adding those amino acids on, you're going to get that polypeptide forming. Okay, and eventually once you hit a stop codon, okay, so you're going to hit a stop codon. There's 3 different stop codons that can be on the mRNA molecule. That's going to tell it to stop. Basically, it's going to cause everything to dissociate. So a release factor, which is a term that you should be familiar with, but you don't need to memorize or anything, will basically tell everything to stop. It'll tell the ribosomal complex, so the ribosomes, to break apart. Okay, and then it'll tell the mRNA strand to be let go. And basically all these things that have been working will now go away. Okay. So this is the overall process of translation. Remember that you have 2 different kinds of RNA involved here. You have the mRNA transcript that we made from the DNA, and then we have the tRNA, which is responsible for bringing the amino acids in that will eventually become the polypeptide. And so if I were to break translation down into steps, as your book does, it talks about initiation, which is a term we've seen before, which is basically when everything comes together at a start codon, so this AUG. Then there's elongation. That's where we start to elongate the polypeptide chain as the ribosome starts moving to the right in this picture relative to the mRNA strand. And then there's finally termination. So, of course, when you are terminated, you are let go or you are stopped, right? And so you hit a stop codon, and that will tell everything to go away. And then you finally have your final protein. And we're going to practice this. We're going to model this. So I don't expect you to be experts yet. But this is just a friendly reminder that all of your traits, so all of your phenotypes, your physical traits, are determined by proteins, right? And so all of this stuff that we did, your cells do a million times over. Okay, this is how we take the information in your DNA and we turn it into you. This is how you turned into you, by making all these different proteins in all the right places according to what your DNA says to do. And so this activity can be enzymatic reactions. So you could be making enzymes to do things. It could be moving proteins around. It could be synthesizing new things, whether it be new molecules, new pigments, anything, or it could be degrading or breaking down stuff. Okay? And so there are a lot of things that can happen if your DNA sequence is messed up. If your DNA sequence is messed up, that might change the protein that you make. And so we're going to practice what happens with mutations, right? Like, will a mutation cause your proteins to be messed up? Sometimes. It depends on the mutation. Okay. And so sometimes you're going to get traits that are abnormal, right? So like that's what's happening down here. If you have a mutation in the PAX6 gene, your eyes will kind of get this dark, cloudy look to them, right? Where you can't quite see the pupil. Here are some extra resources on DNA transcription and translation. I highly recommend that you watch this process in action. I'm going to hope to find some class time to show you what it looks like in action, because it is really involved. There's a lot of moving parts. And so learning to try to recognize pieces of it that you do know is really, really useful. So with that, next time we're going to be talking about gene expression regulation. So basically, like, when do we want to make proteins? Do we always want to make proteins? Because it takes a lot of energy. You don't always want to make proteins. Sometimes you want to calm down. Okay, and so that's going to be where we're going with chapter 18.